Hi, my name is Adrienne Keller, and I'm a postdoc researcher working with Dr. Sarah Hobby. Being a postdoc working at Cedar Creek feels a bit like a homecoming, and as though my research career is coming full circle. As an undergraduate, I had the fantastic opportunity to participate in field and lab research at Cedar Creek. That was my first foray into ecosystem ecology research, and honestly, I've been hooked ever since. As a graduate student at Indiana University, many of the foundational studies I read relating to community and ecosystem ecology came out of research at Cedar Creek. Having research experience under my belt at the revered Cedar Creek Research Station really made me feel connected to the broader ecology research community. And I think this is an important point of the role of Cedar Creek in the past, the present, and the future of the field of ecology. For me, and I think for many others, science and academia can be a bit intimidating and honestly at times alienating. And so for me, as a young female scientist, my connection to Cedar Creek played a small, but I would say not insignificant role in welcoming me into the ecology community. Humans have and will continue to disrupt nutrient cycling across the globe, including in grassland ecosystems, which make up about a third of non-agricultural land globally. As part of the Nutrient Network project, I'm working with Cedar Creek researchers to understand how continued alterations of nutrient loading in grasslands will affect plants and soils. So, for example, the sequestration of carbon in soils is viewed as a potentially powerful climate change mitigation strategy, given that more carbon is stored in soils than vegetation and the atmosphere combined. However, it is unclear how nutrient pollution affects the capacity of these soils to sequester carbon in temperate grasslands. And so to work on this problem, I'm using empirical data from Cedar Creek alongside data from 8 other nutrient network sites to examine how above and below ground plant properties, as well as soil carbon pools, respond to added nitrogen and phosphorus. And the experimental design we use across all 9 sites, including Cedar Creek, is a full factorial fertilization experiment with plots that have been fertilized with nitrogen, with phosphorus, with nitrogen and phosphorus together, as well as control plots, replicated in a blocked design. And after 10 years of fertilization, overall, we find that while fertilization generally stimulates above-ground plant biomass across these sites, we see much more variable effects below ground, with root biomass and root productivity, as well as soil carbon storage, exhibiting negative at some sites, null, and positive responses at other sites to the nitrogen or phosphorus fertilization, depending on the site. And so to me, this highlights the importance of long-term and in-depth site-specific research at Cedar Creek that we can then combine with cross-site analyses, as is possible with Cedar Creek's involvement with the nutrient network.